Good evening and welcome to Dreamy Academy. Tonight we drift far back through time, not to the distant stars but to the hidden worlds beneath our feet, where life weaves itself into an unseen tapestry of profound wonder. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Right now, as you listen, forces older than mountains are shaping the atoms in your bones, and processes more ancient than the first forests are unfolding in the soil beneath your home. We often think of the living world in terms of what we can see, the towering trees, the vibrant flowers, the creatures that walk, swim or fly. But beneath this visible tapestry lies another, far vaster, often silent and endlessly patient kingdom, a realm that underpins nearly all life on Earth, yet remains largely hidden, a silent architect of ecosystems. This hidden kingdom, the fungi, is a subject that rewards our quiet contemplation with a beauty both delicate and immense, a depth that stretches back to the earliest whispers of life on land. It invites us to consider the interconnectedness of all things, the subtle dance of decay and renewal, and the profound wisdom embedded in life's most unassuming forms. To understand fungi is to understand a fundamental pulse of our planet, a rhythm of existence that is both ancient and utterly essential. It is a journey into a world that is not merely strange but utterly beautiful in its quiet complexity, a gentle unravelling of truths that will lull your mind with their intricate logic and their pervasive wonder. Have you ever walked through a forest after a gentle rain, when the air is cool and moist, carrying the scent of damp earth and decaying leaves? Perhaps you've seen a mushroom pushing its way through the soil or emerging from the side of a fallen log, a sudden ephemeral burst of form and colour. We tend to think of this mushroom as the organism itself, a solitary fleeting thing, but that visible cap and stem, however elegant or peculiar, is merely the brief flowering, the quiet declaration of a much larger, far more ancient entity that lies almost entirely out of sight. It is a whisper of a vast, unseen kingdom, unfolding its silent work beneath the ground, within the wood, and through the very fabric of life. Let's begin by considering this hidden truth. The mushroom is not the whole story. It is the fruit, the seed-bearer, of an organism whose true body is an intricate network of fine threads, stretching subtly through its environment. This network, called the mycelium, is the living essence of the fungus. Picture, if you will, an immense subterranean web, woven from countless microscopic strands, each one thinner than a human hair, yet collectively capable of encompassing vast stretches of forest floor or saturating an entire fallen tree. This is where the fungus truly lives, explores, and breathes, a patient, silent intelligence constantly sensing, secreting, and absorbing, connecting itself to everything around it. For a long time, fungi were classified as plants, a simple misunderstanding based on their rooted, stationary nature. But the truth, revealed through patient observation and the quiet language of molecular biology, is far more fascinating. Fungi are not plants. They are not animals either. They are a kingdom entirely unto themselves, as distinct from a tree as a tree is from a human. Their cells, like ours, are eukaryotic, meaning they possess a true nucleus and other complex internal structures, setting them apart from simpler bacteria. But unlike plants, fungi do not photosynthesize. They do not draw energy directly from the sun, and unlike animals, they do not ingest their food internally. Instead, they digest their food externally, secreting powerful enzymes into their surroundings, breaking down complex organic matter into simpler compounds, which they then absorb through their cell walls. This unique mode of feeding is a key to their profound ecological power. Imagine this unseen body, the mycelium, as a vast living exploration system. Each thread, or hypha, pronounced haifa, grows at its tip, extending outward like a patient root, constantly branching, weaving through soil particles, around decaying roots, and into the microscopic crevices of its world. These hyphae are encased in cell walls made primarily of chitin, the same tough, resilient material that forms the exoskeletons of insects and crustaceans. This ancient substance grants the mycelium both flexibility and strength, allowing it to penetrate and persist in environments that would be inhospitable to other forms of life. As these hyphae grow, they form an ever-expanding intricate labyrinth, a silent subterranean architect reshaping the very ground beneath our feet. The sheer scale of these networks can be astonishing. While individual hyphae are tiny, their collective mass and reach can be immense. The largest known organism on Earth by area is a single honey fungus mycelium in Oregon's Malheur National Forest, stretching over 2,200 acres and estimated to be thousands of years old. This vast living network, largely hidden from view, constantly explores its environment, seeking out new sources of nutrients. It is a slow, quiet conquest, not of force but of patient, persistent growth, an unfolding of biological will across the landscape. The growth of a mycelium is not like the growth of a tree reaching for the sky. It is a diffuse, pervasive spread, a gentle infiltration that seeks to connect and transform. This hidden world has been at work for an immense span of geological time. The earliest fossil evidence of fungi dates back over 1 billion years, suggesting they were among the very first complex life forms to emerge. And when life began its tentative steps onto land some 500 million years ago, fungi were there, paving the way. They formed crucial partnerships with the earliest plants, helping them to extract nutrients from barren rock and soil, essentially terraforming the primeval earth. Without these ancient fungal allies, the land might have remained a stark, lifeless expanse. The forests we walk through today, the fertile soils that nourish our crops, and indeed the very atmosphere we breathe, all bear the silent, indelible mark of fungal activity stretching back through eons. They are the quiet, persistent pulse of decomposition and renewal, an invisible engine driving the cycles of life and death. There is a particular comfort in understanding that beneath the fleeting beauty of a mushroom, there exists this ancient, vast and patient intelligence. It changes how we perceive the ground we walk on, transforming it from inert dirt into a teeming, interconnected universe. This underground network, tirelessly breaking down the old to make way for the new, is not just a biological process, it is a profound principle of existence. It tells us that even in decay, there is a vibrant and essential activity, a constant recycling of matter and energy that sustains everything. The universe, it seems, has a tendency toward this elegant efficiency, where nothing is truly lost, only transformed and repurposed. So we've seen that the mushroom is but a fleeting face of a much larger, more enduring organism, the mycelium, a vast hidden network that has quietly shaped our world for billions of years. But this raises a quieter question. How does this silent explorer actually sustain itself? What are the various ways this ancient kingdom interacts with its environment, beyond simply decomposing the fallen? How does it secure the nourishment needed to weave its immense networks, and occasionally send forth its ephemeral fruiting bodies? That question leads us into the diverse and often surprising strategies fungi employ to gather their sustenance, a journey into the many roles they play in the intricate dance of life on Earth. Having journeyed into the unseen architecture of the mycelial whisper, we are left with a profound question, are we not? If the mushroom is but an ephemeral fruit, and the true fungal body, the mycelium, is a vast subterranean intelligence, how does this silent explorer sustain itself? What diverse strategies does it employ beyond the simple act of decomposition to weave its intricate existence into the very fabric of life on Earth? Let us delve deeper into this mystery, allowing our minds to wander through the hidden mechanisms by which fungi draw sustenance, for in understanding their methods, we begin to comprehend the immense quiet power they wield. We discover that the fungal kingdom, far from being a monolithic entity, showcases an astonishing array of trophic strategies, each a testament to evolutionary ingenuity and the profound interconnectedness of all living things. At its most fundamental, we often think of fungi as nature's quiet alchemists, the Earth's patient recyclers. And indeed, a vast number of fungi fall into the category of saprophytes, or decomposers. These are the silent custodians of our forests, the unseen agents transforming fallen leaves, dead wood, and the decaying remains of plants and animals into the very nutrients that nourish new life. Imagine, if you will, a towering oak brought low by a storm. To our eyes, it is a monument of decay, but to the saprophytic fungi, it is a veritable feast, a sprawling larder of cellulose and lignin. Their hyphae, those microscopic threads we spoke of, infiltrate every crevice, every fibre, secreting powerful extracellular enzymes that break down these complex organic compounds into simpler molecules. It is an external digestion, remember, a slow, quiet conquest of matter, absorbed directly through the hyphal cell walls. Without these tireless workers, our planet would be buried under millennia of undecomposed organic matter, its vital nutrients locked away, inaccessible. The cycle of life as we know it would grind to a halt. They are the essential returners, ensuring that what has lived can feed what is yet to come, a profound principle of existence enacted with quiet grace. But the fungal repertoire extends far beyond this crucial role. Some fungi, in a more direct and often dramatic interaction, adopt a parasitic lifestyle. Here the fungus lives on or within another living organism, drawing nutrients from its host, often to the host's detriment. We see this in the plant world, where various fungal pathogens can cause devastating diseases, from mildews that dust grapevines to blights that wither crops. It is a delicate balance, an ongoing evolutionary dance between host and pathogen, where adaptations emerge on both sides. Yet even in these interactions, there is a profound ecological lesson. Parasites often play a role in regulating populations, ensuring biodiversity by preventing any one species from dominating. Consider for a moment the chilling elegance of cordyceps in the infamous zombie ant fungus. This is not a tale of horror, but a testament to specialized adaptation. Imagine an ant foraging in the humid forest floor, unknowingly brushed by a microscopic spore. The spore germinates, its hyphae invading the ant's body, slowly taking control of its nervous system. The ant, compelled by an unseen force, climbs to a high perch, clamps its mandibles onto a leaf or stem, and there it dies. From its head a stalk emerges, a fruiting body poised to release a new shower of spores, ensuring the continuation of the fungus. It is a stark, captivating example of a parasitic strategy, illustrating the astonishing lengths to which life will go to find its niche. This is not malevolence, but simply a strategy of survival, a fascinating if somewhat unsettling chapter in the fungal story. However, it is perhaps in the realm of a mutualism, where fungi truly reveal their most astonishing and widespread strategies, forming partnerships that are not only beneficial, but often absolutely essential for the survival of their hosts, and indeed, for entire ecosystems. These are the collaborations that underscore the interconnectedness of all things, where two disparate life forms come together to create something greater than the sum of their parts. Foremost among these are the mycorrhizal fungi, a term derived from the Greek words for fungus and root. This ancient partnership, dating back hundreds of millions of years, is believed to have been absolutely critical for plants to colonize land. We know that plants with their roots can absorb water and nutrients from the soil, but fungal hyphae, being far finer and more extensive than any plant rootlet, can explore a vastly larger volume of soil. Imagine the sheer scale, an immense subterranean web, an underground internet of fungal threads, connecting trees and plants across vast stretches of forest. In this profound exchange, the plant, through photosynthesis, produces sugars, the very energy of life. It then shares a portion of these precious carbohydrates with its fungal partner. In return, the fungus, with its unparalleled ability to extend into the soil and secrete enzymes, acts as an incredibly efficient scavenger. It dramatically increases the plant's access to water and vital nutrients, especially phosphorus and nitrogen, which are often scarce or locked away in forms inaccessible to plant roots alone. The fungus effectively extends the plant's root system by orders of magnitude, protecting it from pathogens and even helping it withstand drought. It is a silent, tireless negotiation, unfolding beneath our feet, a testament to the power of cooperation. When we walk through a forest, we are not just seeing individual trees, we are witnessing a vast interconnected community, largely orchestrated by this patient, silent intelligence of the mycorrhizal network. Then there are the lichens, those enigmatic, crusty, leafy or shrubby growths we often see clinging to rocks, tree bark and even barren tundra. These are not single organisms but composite entities, a profound marriage of kingdoms. A lichen is typically formed by a fungus, the mycobiont, living in intimate association with an alga or a cyanobacterium, the photobiont. The fungal partner provides structure, protection from desiccation and UV radiation, and absorbs water and minerals from the environment. The algal or cyanobacterial partner, in turn, performs photosynthesis, producing sugars that feed both itself and the fungus. Lichens are true pioneers, capable of colonizing extreme environments where no other life can survive, slowly breaking down rock and creating the first rudimentary soils, paving the way for future plant life. They are living symbols of resilience and collaboration, embodying the wisdom of shared existence in the harshest of conditions. And the partnerships continue. We find endophytic fungi, which live entirely within the tissues of plants, often without causing disease. These microscopic residents can provide their hosts with a range of benefits, from enhanced resistance to pests and pathogens to increased tolerance to environmental stresses like drought or heavy metals. It is a hidden layer of protection, an internal guardian, showcasing the subtle and often unseen ways in which fungi support plant life. Even more astonishingly, some fungi exhibit predatory behaviors. Yes, you heard correctly. There are predatory fungi that have evolved specialized structures to trap and consume tiny organisms, particularly nematodes, microscopic roundworms that inhabit the soil. Some develop constricting rings that inflate rapidly when a nematode passes through, snaring the unsuspecting worm. Others produce adhesive knobs or networks of sticky hyphae that ensnare their prey. Once trapped, the fungus penetrates the nematode's body and digests it from within. These are the tiny subterranean hunters, adding another layer of wonder to the fungal kingdom's diverse strategies. What we witness in these varied nutritional modes is not merely a collection of biological curiosities, but a profound illustration of life's adaptability and its unwavering drive to persist. From breaking down the old to building new alliances, from subtle internal partnerships to direct predation, fungi demonstrate an unparalleled flexibility in their relationship with the living world. They are not simply passive recyclers, they are active participants, shaping ecosystems, driving nutrient cycles, and fostering the very conditions that allow other life forms to thrive. As we reflect on these diverse strategies, we begin to appreciate the immense quiet intelligence embedded in these unassuming forms. They are the essential threads in the unseen tapestry of life, constantly negotiating, competing, and collaborating. They remind us that survival often lies not in isolation, but in connection, in the intricate dance of giving and taking that defines all existence. But how do these silent architects, with all their varied methods of sustenance, impact the world on an even grander scale? Beyond their individual interactions, what is their collective ecological footprint? And how do they contribute to the broader health and resilience of our planet's biomes? This is a question that invites us to broaden our perspective, to move from the microscopic interactions to the macroscopic consequences, as we continue our slow and thoughtful adventure into the mycology fundamentals. We have journeyed through the intricate tapestry of fungal life, from the unseen network of mycelium to the diverse strategies by which these organisms sustain themselves. We have marveled at their ancient origins, their unique methods of digestion, and the myriad relationships they forge, from the silent work of saprophytes to the profound partnerships of mycorrhizae and lichens. Yet, as we concluded our last contemplation, a grander question began to unfurl in our minds. What is the macroscopic ecological impact of this hidden kingdom? How do these silent architects, these patient explorers, influence the very health and resilience of our planet on a scale far vaster than a single forest floor or a decaying log? Let us now widen our gaze, you and I, to encompass the immense pulsating systems of Earth itself, and consider how fungi are not merely participants, but fundamental operators within these global cycles. We often speak of the web of life, a phrase that, while beautiful, perhaps doesn't fully capture the sheer depth and interconnectedness of biological processes. If life is a web, then the fungal kingdom, with its immense subterranean networks, its intricate biochemical pathways, and its relentless, quiet work, is surely a foundational strand, perhaps even the very nervous system that transmits and processes the planet's vital information. Consider the vastness of organic matter that falls to Earth each day. Leaves shed from countless trees, branches broken by storms, the bodies of animals that have completed their life's journey. Without a robust and efficient system of decomposition, our planet would quickly become suffocated under its own detritus. This is where fungi, particularly the saprophytes we discussed, emerge as the planet's most diligent and tireless recyclers. They are not merely breaking down waste, they are dismantling complex organic structures, atom by atom, molecule by molecule, and returning those precious building blocks, carbon, nitrogen, phosphorus, potassium, back to the soil, where they can be reabsorbed by plants, thus fueling new life. It is an immense, ceaseless alchemy transforming death into the potential for rebirth. Imagine, if you will, the sheer volume of carbon locked within a single tree or an entire forest. When that tree dies, that carbon, along with other essential nutrients, remains sequestered, unavailable. Fungi are the primary agents capable of breaking down lignin and cellulose, the tough recalcitrant polymers that give wood its strength. Without them, forests would pile up, carbon would remain trapped, and the global carbon cycle, a delicate dance between atmosphere, oceans, and living organisms, would grind to a halt. We often focus on the grand photosynthetic capture of carbon by plants, but it is the fungal release of that carbon in a controlled and cyclical manner that keeps the entire system in dynamic balance. They are, in essence, the planet's slow-release mechanism, ensuring that life's essential elements are never truly lost, only transformed and recirculated. This tireless work extends far beyond the visible act of decay. Beneath our feet, the mycelial networks are not just breaking down matter, they are literally building the soil itself. Think of soil not as inert dirt, but as a living, breathing entity, a complex matrix teeming with life. Fungal hyphae weave through the soil particles, binding them together, creating stable aggregates. This aggregation is crucial for soil structure, preventing erosion by wind and water and creating pore spaces that allow for aeration and water infiltration. A healthy, fungal-rich soil acts like a sponge, holding moisture, preventing runoff, and making nutrients more accessible to plants. In this way, fungi are truly the architects of fertile ground, sculpting the very foundation upon which terrestrial ecosystems thrive. They create the stable, vibrant substrate that allows roots to anchor, water to flow, and life to flourish. Without this unseen, intricate engineering, much of the land we stand upon would be barren, washed away by the slightest rain. And what of the broader implications for our climate, for the very air we breathe? The fungal kingdom plays a profound, albeit often overlooked, role in regulating the planet's carbon balance. As we just explored, their decomposition of organic matter releases carbon back into the atmosphere, but their role is far more nuanced than simple release. Mycorrhizal fungi, through their vast networks, can significantly influence how much carbon is stored in the soil. They facilitate the transfer of carbon-rich compounds from plants to the soil, where it can be stabilized and sequestered for centuries, even millennia. The sheer biomass of fungal hyphae beneath our feet represents an immense carbon sink, a living reservoir that holds vast quantities of this element, preventing its rapid return to the atmosphere as CO2. They are quiet guardians of the carbon cycle, moderating its flow, influencing the very composition of our atmosphere. Moreover, consider the resilience of ecosystems. When a forest fire sweeps through a landscape or a clear-cut devastates a patch of land, the initial impression is often one of utter destruction. But beneath the charred surface or within the disturbed earth, the fungal networks often persist, dormant yet ready. They are among the first responders in ecological succession, aiding pioneer plants in recolonizing barren ground, facilitating nutrient uptake in depleted soils, and accelerating the healing process. They are the patient healers, the silent infrastructure that allows life to rebuild, to adapt, and to regenerate. This capacity for resilience, this inherent drive towards equilibrium and renewal, is deeply embedded within the fungal kingdom. The concept of the wood wide web, which we touched upon briefly when discussing mycorrhizal fungi, extends beyond individual trees and their fungal partners. It speaks to a planetary scale network of communication and resource distribution. Imagine the Earth's terrestrial biomes interconnected by these vast subterranean mycelial threads, a slow biological internet transmitting nutrients, water, and even chemical signals across vast distances. Forest trees, even those of different species, can share resources through these networks, providing support to struggling saplings or distributing nutrients from areas of abundance to areas of scarcity. It is a profound demonstration of interconnectedness, a living testament to the idea that no organism truly exists in isolation. This immense living network fosters a collective intelligence, a shared resilience that allows entire ecosystems to function as integrated, adaptive wholes. As we reflect on these grand contributions, we begin to see fungi not just as fascinating biological curiosities, but as indispensable partners in maintaining planetary health. They are the unsung heroes of decomposition, the quiet architects of soil, the subtle regulators of climate, and the resilient engineers of ecosystem recovery. Their presence, often unseen and unappreciated, underpins the very stability and vitality of life on Earth. They remind us that the most profound forces are often the most subtle, working tirelessly beneath the surface, shaping our world in ways we are only just beginning to comprehend. We stand at the precipice of a deeper understanding, don't we? An understanding that invites us to move beyond a human-centric view of life, to embrace the profound wisdom embedded in life's most unassuming forms. The fungal kingdom, with its ancient lineage and its ceaseless quiet work, offers us a powerful lesson in interconnectedness, resilience, and the delicate balance required for sustained existence. It teaches us that true strength often lies not in dominance but in collaboration, in the intricate dance of giving and receiving that sustains the whole. And as we conclude this exploration of their macroscopic impact, we are left with a renewed sense of awe for these ancient, patient, and utterly essential inhabitants of our shared planet. The question now, as our journey continues, is not merely what they do, but how they achieve such wonders at a cellular and structural level. How are these magnificent networks built, and what forms do they take? We have journeyed through the vast, silent history of fungi, witnessing their ancient origins, their foundational role in life's grand tapestry, and the astonishing diversity of their ecological strategies. We have marvelled at their tireless work as decomposers, their intricate partnerships, and their profound influence on the very fabric of our planet, from healthy soil to climate regulation. Yet, as we concluded our last exploration, a fundamental question lingered, hovering like mist over a forest floor. How do these unseen architects achieve such intricate wonders? What are the specific forms their magnificent networks take, and what secrets lie within their cellular structures that enable such silent, profound intelligence? Today we delve deeper, moving from the grand macroscopic narrative to the exquisite microscopic realm. We invite you to imagine shrinking down not merely to the size of a fallen leaf, but to a scale where individual threads become vast pathways, and a single cell holds an entire universe of purpose. For it is here, within the very architecture of the fungal body, that we begin to truly grasp the genius of this kingdom. At its most fundamental level, the fungal organism is built upon a deceptively simple yet profoundly elegant structure, the hypha. Plural. Hyphae. Imagine, if you will, an infinitely branching microscopic thread, a delicate filament that is the very essence of fungal growth and exploration. But before we trace the journey of these threads, let us first peer into the individual building blocks that comprise them, the fungal cells themselves. Unlike the neatly compartmentalized cells of animals, or the rigid cellulose-walled cells of plants, fungal cells possess a unique character. They are first and foremost eukaryotic, meaning they house a true nucleus and other membrane-bound organelles like mitochondria, the powerhouses of the cell, and vacuoles, which store nutrients and maintain turgor pressure. In this, they share a kinship with us, a distant echo of a shared evolutionary past. However, their most defining feature, the very armour that protects and shapes them, is their cell wall. Where plants rely on cellulose for structural integrity, fungi construct their walls primarily from chitin. Think of chitin as nature's incredibly strong, flexible, and resilient biopolymer. It is the same substance that gives insects their hard exoskeletons and crabs their sturdy shells. This chitinous armour provides immense strength and rigidity, allowing fungi to exert the necessary pressure to penetrate substrates, to withstand osmotic stress, and to maintain their delicate tubular forms. It is a testament to nature's efficiency, a profound example of convergent evolution, where distinct lineages arrive at similar solutions for similar challenges. We often overlook such details, yet it is in these microscopic differences that entire kingdoms diverge, carving their unique paths through the biosphere. Now, let us return to the hypha. A single hypha is essentially a tubular cell or a series of cells, encased in this chitinous wall. But here, another fascinating divergence appears. Many fungal hyphae are divided by internal crosswalls called septa, singular cell, septum. These septa, however, are rarely complete. Instead, they are typically perforated by a central pore, or sometimes multiple pores, allowing the cytoplasm, nutrients, and even nuclei to flow freely from one compartment to the next. This creates a remarkable continuity, a living pipeline where resources can be rapidly mobilized across vast distances within the mycelial network. This continuous flow is a profound principle, a physical manifestation of the interconnectedness we have discussed, allowing the entire organism to act as a single coordinated entity, even when spread across meters or even kilometers of substrate. Other fungi, particularly some of the more ancient lineages, possess coenocytic or aseptate hyphae. In these forms, there are no septa at all. The hypha is a continuous multinucleate tube, a true syncytium. Imagine a single elongated cell containing hundreds or thousands of nuclei all sharing a common cytoplasm. This too speaks to a different kind of efficiency, a rapid, unrestricted flow of resources and genetic material throughout the entire organism. Whether septate or coenocytic, the underlying principle remains: the fungal body is designed for rapid growth, efficient transport, and pervasive exploration. The growth of a hyphae is a marvel of biological engineering, a process known as apical growth. The hyphal tip is the engine of exploration, a highly dynamic and sensitive structure. Here, the cell wall is more pliable, allowing for expansion. Vesicles, tiny membrane-bound sacs filled with enzymes and cell wall precursors, are continuously transported to the very tip. These enzymes are then secreted externally, breaking down the surrounding substrate, while new cell wall material is simultaneously incorporated, pushing the tip forward. It is a slow, quiet conquest, a relentless probing of the environment. Think of it as a microscopic, intelligent drill, constantly extending, constantly seeking out new resources, new pathways. As the hyphae grows, it also branches. These branches arise either from the main hyphal axis or from existing branches, creating an ever-expanding intricate web. This branching pattern is not random. It is an intelligent response to the environment, dictated by nutrient gradients, moisture levels, and the presence of obstacles. The hyphae literally sense their surroundings, growing towards sources of food and water, navigating around barriers, and optimizing their search strategy. It is this continuous, directed apical growth and branching that gives rise to the true heart of the fungus, the mycelium. The mycelium is the collective mass of these individual hyphae, an immense subterranean web, often hidden from our view. When we speak of the true organism of the fungus, it is the mycelium we refer to. The mushroom, that ephemeral burst of form and colour we so often admire, is merely the reproductive structure, the fruit of this vast hidden network. Imagine the mycelium as a vast living neural network, spread throughout the soil, wood or any other substrate it colonizes. Each hypha is a thread, a conduit, a sensory probe. Together they form a complex, interconnected system capable of immense feats. The sheer scale of some mycelial networks is truly astonishing. We have spoken of Armillaria ostiae, the honeycomb mushroom, whose mycelial network in Oregon spans over 200 acres and is estimated to be thousands of years old, making it one of the largest and oldest living organisms on Earth. To comprehend this is to stretch our imaginations, to grasp the profound principle of existence that operates largely beyond our direct perception. It is a testament to the quiet power of cumulative microscopic growth. The mycelium's primary function is absorption. As the hyphal tips extend, they secrete a cocktail of powerful extracellular enzymes directly into their surroundings. These enzymes break down complex organic molecules, the lignin and cellulose of wood, the proteins and carbohydrates of dead organisms, into smaller absorbable units. These simpler molecules are then taken up through the hyphal cell walls and transported throughout the entire network via the cytoplasmic streaming through those septal pores. This external digestion is a fundamental difference from animals, which ingest and then digest internally, and from plants, which photosynthesize. It is a highly efficient and adaptable strategy, allowing fungi to thrive on an incredible diversity of substrates. But the mycelium is not merely a passive, absorptive mat. It is a dynamic, adaptive structure. It can differentiate into various specialized forms, each serving a specific purpose, each a further testament to the profound wisdom embedded in life's most unassuming forms. Consider, for instance, rhizomorphs. These are not individual hyphae, but highly organized macroscopic bundles of hyphae, resembling tiny roots or shoelaces. Within a rhizomorph, the hyphae are tightly packed, often differentiated, with an outer protective layer and an inner core designed for efficient long-distance transport of water and nutrients. Rhizomorphs are the fungus's highways, its superhighways, allowing it to bridge gaps in substrates, explore new territories far from its initial point of colonization, and even penetrate tough materials like bark or compact soil. They are a visible manifestation of the mycelium's exploratory drive, its sullen, patient intelligence seeking to expand its dominion. Have you ever lifted a piece of decaying wood in a damp forest and seen dark, thread-like structures running beneath it? Those are often rhizomorphs, the silent explorers charting new courses through the hidden world. Then there are sclerotia, singularly sclerotium. These are compact, hardened masses of mycelium, often rounded or irregularly shaped, acting as survival structures. Imagine them as tiny living time capsules. When environmental conditions become harsh, perhaps too dry, too cold, or lacking in nutrients, the mycelium can consolidate its resources, forming these dense, resistant structures. Sclerotia are incredibly resilient, capable of remaining dormant for extended periods, sometimes years, only to germinate and resume active growth when favourable conditions return. They are a profound example of fungal resilience, a testament to their ancient ability to weather environmental fluctuations and persist through time, silently waiting for the opportune moment. Beyond these, we find other specialized forms: fungal cords and strands, which are less organized bundles of hyphae, often found spreading through decaying wood. Haustoria, specialized hyphal structures formed by parasitic fungi to penetrate and absorb nutrients directly from host cells, and the intricately branched weae, arbuscules, formed by mycorrhizal fungi within plant root cells, facilitating the crucial exchange of nutrients. Each of these forms, from the simplest hyphal tip to the complex rhizomorph, represents an elegant adaptation, a refined solution to the challenges of survival and growth in a competitive world. The interconnectedness within the mycelial network is not just about nutrient transport, it's also about communication. Chemical signals can travel throughout the network, allowing the fungus to coordinate its growth, respond to threats, and even share resources. If one part of the mycelium encounters a rich food source, that information can be relayed to other parts, directing growth towards it. If another part is under attack, defenses can be mobilized across the entire system. This distributed intelligence, this ability for the whole to act as more than the sum of its parts, is what makes the mycelium such a powerful and pervasive force in ecosystems. It is truly the wood wide web at its most fundamental structural level. As we conclude this exploration of the fungal blueprint, we are left with a deeper appreciation for the profound elegance of its design. From the chitinous strength of its cell walls to the relentless probing of its hyphal tips and the vast intricate networks of its mycelium, every aspect speaks to an ancient wisdom, a perfected strategy for life. The fungus, through its hidden architecture, reminds us that the most powerful forces are often the most unseen, the most patient, and the most interconnected. It is a quiet testament to the immense creativity of nature, a silent architect continually reshaping the world beneath our feet. But what happens when this cell's hidden network decides it is time to reveal itself, however briefly? What are the mechanisms by which this our subterranean intelligence culminates in the ephemeral beauty of a mushroom, a puffball, or a bracket fungus? How does the mycelium with its endless threads orchestrate the formation of these complex reproductive structures? This perhaps is where our next journey will lead us, into the visible world of fungal fruiting and the fascinating strategies of reproduction. As we've journeyed deeper into the unseen architecture of the fungal kingdom, we've explored the silent, relentless expansion of the mycelial network, a vast subterranean consciousness, tirelessly digesting, connecting, and sustaining life. We've marveled at its cellular blueprint, the seemingly simple yet profoundly effective hypha, and the collective intelligence of the mycelium. But a question perhaps lingers in the quiet corners of your mind, a natural curiosity born from observing the occasional ephemeral emergence of these hidden beings. How does this immense, patient, and largely unseen web orchestrate the sudden, often spectacular appearance of the mushroom? What intricate strategies do these ancient organisms employ to bring forth the next generation, to cast their future into the wind? This brings us, dear friends, to the fascinating, often beautiful, and endlessly diverse world of fungal reproduction. It is here that the unseen becomes visible, if only for a fleeting moment, a testament to life's persistent drive to perpetuate itself. The mushroom, that familiar cap and stem, is not the fungus itself, but rather its reproductive organ, a complex and highly specialized structure designed with one primary purpose: to produce and disperse spores. It is the flag unfurled for a brief, glorious period, signaling the success of the unseen mycelium beneath. Imagine, if you will, the immense, quiet energy gathered within the mycelial network, a slow, deliberate accumulation of resources and genetic information, all building towards this singular, crucial moment. The formation of a mushroom, or any fungal fruiting body, is a marvel of biological engineering, a sudden burst of organized growth from an otherwise diffuse and sprawling organism. It is a testament to the mycelium's capacity for differentiation, its ability to shift from a uniform, exploring network to a highly structured, specialized form. Environmental cues, changes in temperature, humidity, light, or nutrient availability, often trigger this monumental shift, signaling that the conditions are ripe for the delicate process of reproduction. Fungi, in their ancient wisdom and incredible adaptability, have evolved a breathtaking array of reproductive strategies, a tapestry woven with threads of both elegant simplicity and astonishing complexity. These methods ensure their survival across an astonishing diversity of habitats, from the deepest oceans to the highest mountains, from the arid desert to the dampest rainforest. We can broadly categorize these strategies into two fundamental paths: asexual and sexual reproduction. Both are vital, each offering distinct advantages in the grand, ongoing saga of life. Let us first consider the path of asexual reproduction, a strategy centered on efficiency, speed, and the faithful replication of successful genetic blueprints. Here, the fungus essentially clones itself, producing offspring that are genetically identical to the parent. This can be achieved through several elegant mechanisms. Perhaps the simplest is fragmentation, where a piece of the mycelium breaks off, either accidentally or intentionally, and, if conditions permit, grows into a new, independent colony. It's a testament to the inherent resilience and regenerative capacity of the hyphae, each segment holding the potential for an entire new organism. Then there is budding, a common method for single-celled fungi like yeasts. Picture a small bulge forming on the surface of the parent cell, growing larger and eventually pinching off to become a new, genetically identical yeast cell. It's a rapid, unassuming process, yet incredibly effective for quick colonization and nutrient exploitation, allowing yeast to proliferate swiftly in sugar-rich environments like the fermenting juices we harness for bread and wine. However, the most widespread form of asexual reproduction in many fungi involves the production of spores. These are specialized reproductive cells, often microscopic, designed for dispersal and survival. In many fungi, particularly the molds we might observe on forgotten food, asexual spores are produced in vast numbers. Think of the fluffy green or black patches on bread or fruit. These are often masses of conidia, spores produced externally on specialized hyphae, called conidiophores, or sporangiospores, produced internally within sac-like structures called sporangia. These spores are like tiny airborne seeds, each capable of germinating into a new mycelium when it lands in a favorable environment. The sheer volume of these spores, released into the air with every disturbance, speaks to a strategy of overwhelming odds, ensuring that at least a few will find fertile ground. It's a silent, microscopic rain, constantly falling, constantly seeking. Yet while asexual reproduction offers speed and consistency, it lacks the genetic shuffling that fuels evolution and adaptability. For this, fungi turn to the profound and intricate dance of sexual reproduction, a process that introduces genetic variation, allowing populations to adapt to changing environments and exploit new niches. This is where the story of the mushroom truly unfolds in its full complexity. Sexual reproduction in fungi typically involves three key stages. 1. A plasmogamy. The fusion of the cytoplasm of two parent hyphae. Interestingly, the nuclei often do not fuse immediately. 2. Karyogamy. The fusion of the two haploid nuclei from the two parents to form a single diploid nucleus. This is the moment of true genetic recombination. 3. Meiosis. The diploid nucleus then undergoes meiosis, a cell division process that reduces the chromosome number by half, producing genetically diverse haploid spores. It is during this intricate ballet of nuclear fusion and division that the incredible diversity of fungal forms, and indeed the very essence of the mushroom, comes into being. Let us consider the majestic Basidiomycetes, the group that includes most of the mushrooms we recognize, the familiar toadstools, shelf fungi, puffballs and bracket fungi. Here the visible mushroom is known as a basidiocarp, the fruiting body specifically designed to produce and disperse basidiospores. Imagine, if you will, the quiet stirrings within the mycelium, deep beneath the soil or within the heart of a decaying log. When the time is right, often after a rain or a shift in seasons, tiny knots of hyphae begin to organize, forming a primordial structure, a pinhead or button. This small, seemingly insignificant structure, drawing nutrients and water from the vast mycelial network, begins to swell and differentiate with astonishing speed. The cells within it divide and expand, arranging themselves into the distinct parts we recognize: the cap, pileus, the stem, steep, and often a delicate ring, annulus, or a cup-like base, vulva, remnants of the protective veil that once encased the developing mushroom. Beneath the cap, the true magic unfolds, often hidden within the intricate architecture of gills or pores. Here, on the surface of these delicate lamellae, or within the walls of these tiny tubes, are microscopic club-shaped structures called basidia. Each basidium is a biological marvel, a miniature factory where karyogamy and meiosis occur. Two compatible nuclei fuse, creating a temporary diploid state which then quickly undergoes meiosis to produce 4 genetically unique haploid basidiospores. These spores mature and are typically borne externally on tiny stalks, sterigmata, atop the basidium. The dispersal of these basidiospores is a masterclass in passive yet effective engineering. As the mushroom cap expands and lifts, it creates a precise microclimate. The gills or pores are spaced to allow air currents to carry the spores away. Many species even employ an active discharge mechanism, a tiny droplet of water forming and then releasing the spore with a sudden, forceful propulsion, ensuring it clears the gill surface before gravity takes over. The sheer volume is staggering. A single mature mushroom can release billions of spores over its short lifespan, a silent cascade drifting on the breeze, each a potential new life. Then there are the ascomycetes, a vast and diverse group encompassing cup fungi, morphology, truffles, and many yeasts. Their sexual fruiting bodies, known as ascocarps, take on an equally varied and often beautiful array of forms, from the elegant goblet-like cups of Pizziza to the honeycomb-like structures of morels and the subterranean treasures of truffles. Within these ascocarps, the spores are produced inside sac-like cells called asci, singular ascus. In an ascus, after plasmogamy and karyogamy, the diploid nucleus undergoes meiosis, followed by a mitotic division, typically resulting in 8 ascospores neatly arranged within the sac. When mature, these ascospores are often forcibly ejected, sometimes in a visible puff of smoke from the ascus. Imagine walking through a damp forest and seeing a small cup-shaped fungus, and then, with a gentle touch or a sudden shift in humidity, a cloud of microscopic spores is released into the air. It is a subtle, yet powerful act of procreation. Truffles, those highly prized subterranean ascomycetes, employ an entirely different dispersal strategy, a fascinating partnership with the animal kingdom. Their ascocarps develop entirely underground, releasing their pungent, alluring aromas to attract mammals, pigs, dogs, squirrels, who unearth and consume them, inadvertently dispersing the spores through their scat. It is a testament to the intricate web of life, where the survival of one species becomes entwined with the habits of another. Even the humble zygomycetes, like the bread molds we mentioned earlier, engage in sexual reproduction, forming thick-walled, resistant zygospores when two compatible hyphae meet. These zygospores can withstand harsh conditions, lying dormant until favourable circumstances return, a testament to the fungal kingdom's deep-seated resilience. The journey of a spore, once released, is one of immense challenge and profound potential. Blown by the wind, carried by water, or transported by insects and animals, only a minuscule fraction will land in a suitable environment, one with the right temperature, humidity and nutrient availability to germinate. But for those fortunate few, the process begins anew. The spore swells, a germ tube emerges, and from this single delicate thread, a new network of hyphae begins its slow, quiet conquest, extending its unseen tendrils into the substrate, patiently building towards its own ephemeral burst of form and colour. This cycle, from spore to hypha to mycelium to fruiting body and back to spore, is a fundamental rhythm of life on Earth. It speaks of continuity, of an unwavering commitment to existence, even in the face of overwhelming odds. The ephemeral beauty of a mushroom rising from the earth in a single night and fading just as quickly holds within its brief existence the distilled wisdom of eons of evolution. It is a fleeting masterpiece, a testament to the hidden intelligence that guides life's most unassuming forms, ensuring that the silent architects continue their tireless work, perpetually weaving the unseen tapestry of our world. As we conclude this exploration of fungal reproduction, we are left once again with a sense of awe at the sheer ingenuity of life. These microscopic and macroscopic strategies, from the simple budding of yeast, the complex architecture of a mushroom, highlight the profound adaptability and persistent vitality of the fungal kingdom. They remind us that even the most fleeting of appearances can be the culmination of immense, unseen effort, a critical moment in the ongoing interconnected story of our planet. What wonders we might ponder are unfolding beneath our very feet at this precise moment, preparing for their own quiet yet spectacular emergence. We have journeyed together, delving into the unseen architecture of the hypha and the intricate ballet of fungal reproduction, witnessing how life perpetuates itself in myriad ingenious ways. From the simplest fragmentation to the complex choreography of sexual spore formation, we've seen the fundamental mechanisms that ensure the fungal kingdom's enduring presence. But these mechanisms, vital as they are, serve a grander purpose: to manifest an astonishing diversity of forms, strategies and ecological roles that continue to shape our world in ways we are only beginning to truly appreciate. Now, we invite you to broaden our gaze, to step back and contemplate the full, breathtaking spectrum of this kingdom. For while we speak of fungi as a singular entity, it is, in truth, an immense variegated tapestry woven from countless threads, each representing a unique lineage, a distinct evolutionary path, a specialized adaptation to life's endless niches. To truly grasp the profound impact of fungi, we must explore their major divisions, their phyla, these ancient branches on the tree of life, each holding its own secrets and wonders. Our exploration begins, perhaps surprisingly, with groups that often escape the popular imagination, yet are foundational to the fungal story. Consider for a moment the Chytridiomycota, the chytrids. These are, by many accounts, among the most ancient lineages of fungi, a whisper from deep time echoing across the eons. Unlike many of their more familiar relatives, chytrids are largely aquatic, thriving in freshwater and marine environments, as well as damp soils. What sets them apart, what connects them to a primal past, is the presence of motile spores, zoospores, equipped with a single posterior flagellum. Imagine these microscopic tail-waving entities swimming through water, seeking out new substrates or hosts. It's a glimpse into the very origins of fungal life, a reminder of their aquatic ancestry before the grand terrestrial colonization. While many chytrids are simple decomposers, recycling nutrients in aquatic ecosystems, some are also notorious parasites, like Batrachochytrium dendrobatidis, the devastating pathogen responsible for widespread amphibian declines across the globe. This single organism, tiny and seemingly insignificant, holds the power to reshape entire ecosystems, a stark reminder of the delicate balance of life and the potent influence of even the smallest fungal forms. Moving onward, we encounter the Zygomycota, a group now often referred to as Mycorrhizal, representing another ancient lineage, though one more commonly terrestrial. You might know them as the fast-growing molds that appear on forgotten bread, spoiled fruit, or even decaying vegetables, the fuzzy, often dark patches that seem to materialize overnight. These are the work of genera like Rhizopus or Mucor tireless saprophytes, breaking down simple sugars with remarkable efficiency. Their hyphae are typically coenocytic, meaning they lack septa, allowing for a rapid, unrestricted flow of cytoplasm and nutrients throughout the mycelial network, a testament to their explosive growth strategy. Their name, Zygomycota, derives from the characteristic zygospores they produce during sexual reproduction. Imagine two compatible hyphae meeting, their tips fusing to form a robust, thick-walled zygosporangium, a survival capsule capable of enduring harsh conditions, patiently waiting for the return of favourable circumstances before releasing a new generation of spores. It is a strategy of resilience, of patient endurance, a quiet wisdom embedded in their very life cycle. Next, we turn our attention to a group whose members may never form a visible mushroom, yet whose ecological impact is utterly profound: the Glomeromycota. These are the architects of the arbuscular mycorrhizal AM relationship, the very fungi we discussed as the bedrock of the underground internet. Indeed, all known species within the Glomeromycota are obligate symbionts, meaning they cannot complete their life cycle without forming a partnership with a plant host. Their hyphae, once they have explored the soil, penetrate the root cells of vast numbers of plants, forming tree-like structures called arbuscules, intimate interfaces for nutrient exchange. They do not produce elaborate fruiting bodies, but rather large, often multinucleate spores in the soil, which serve to propagate their lineage. We cannot overstate their importance. An estimated 80% of all vascular plants on Earth rely on these ancient partnerships. Without the Glomeromycota, the vast majority of terrestrial ecosystems as we know them simply would not exist. They are the silent, unseen threads that bind the plant kingdom to the soil, facilitating the flow of life-sustaining elements, a testament to the power of cooperation, of mutual benefit, a profound principle woven into the fabric of life itself. Now, we arrive at the two largest and arguably most diverse phyla, groups that encompass much of what we commonly recognize as fungi and so much more. First, let us explore the Ascomycota, often called the sac fungi, a name derived from their distinctive sexual reproductive structure, the ascus. Within these microscopic sac-like cells, typically 8 ascospores are produced, each a tiny vessel carrying the genetic blueprint for a new generation. The Ascomycota are a kingdom within a kingdom, a realm of astonishing variety. Consider the humble yeasts, those microscopic, often single-celled fungi that have partnered with humanity for millennia. Saccharomyces cerevisiae, the baker's and brewer's yeast, is perhaps the most famous silently transforming sugars into alcohol and carbon dioxide, giving us bread that rises and beverages that cheer. It is a simple yet profound alchemy, a testament to the unseen metabolic power of these tiny organisms. But yeasts are far more diverse than this. They inhabit our skin, our guts, and every corner of the environment, playing myriad roles from decomposition to symbiosis. Then there are the molds, many of which belong to the Ascomycota. Think of the genus Penicillium, which gave us penicillin, the miraculous antibiotic that revolutionized medicine, a serendipitous discovery revealing the powerful biochemical arsenal of fungi. Or Aspergillus, a genus vital for producing enzymes, fermenting soy sauce and even citric acid, yet also containing species that can cause serious human diseases or produce dangerous toxins in crops. These filamentous fungi, with their often colourful fuzzy colonies, are tireless decomposers, recyclers and chemical engineers of the microbial world. And of course the Ascomycota give us some of the most prized culinary delights, the earthy, enigmatic truffles, whose subterranean aromas draw animals and gourmands alike, the intricate honeycombed morels, harbingers of spring, and the elegant, often vibrantly coloured cup fungi that dot the forest floor. These are the visible manifestations of vast hidden mycelial networks, their fruiting bodies designed with exquisite precision to disperse their ascospores. Let us not forget the lichens, those pioneering organisms we touched upon earlier. The fungal partner in the vast majority of lichens, a staggering 98%, is an ascomycete. The fungus forms an intimate, enduring partnership with an alga or cyanobacterium, creating a composite organism capable of colonizing the most inhospitable environments, slowly dissolving rock and paving the way for other life forms. It is a profound testament to interspecies collaboration, a living symbol of resilience and adaptation, orchestrated by an ascomycete. Finally, we arrive at the Basidiomycota, often known as the club fungi, a name that evokes the club-shaped Basidia, on which their sexual spores, the basidiospores, are born. This phylum encompasses the vast majority of what the average person recognizes as a mushroom. The classic cap and stem forms, the elegant gills, the sturdy pores, the delicate brackets, and the whimsical puffballs that release clouds of spores with a gentle squeeze. Within the Basidiomycota, we find an incredible array of ecological roles. Many are powerful decomposers of wood, including the formidable white rot fungi, which possess the unique enzymatic machinery to break down lignin. the tough polymer that gives wood its rigidity. Without these basidiomycetes, our forests would be choked with fallen timber, and the global carbon cycle would grind to a halt. They are the silent, tireless architects of decay and renewal, returning vital nutrients to the soil. Others are crucial. Mycorrhizal partners, forming ectomycorrhizal relationships with trees, enveloping their root tips in a fungal sheath and extending vast networks into the soil. Think of the majestic boletes, the vibrant amanita species, or the delicate chanterelles. Many of these are intimately connected to the root systems of specific trees, forming ancient, indispensable partnerships that underpin forest health and vitality. But the Basidiomycota are not solely benevolent. This phylum also includes some of the most economically destructive plant pathogens, such as the war rusts and smuts. These microscopic parasites can devastate agricultural crops, causing billions of dollars in losses annually, a powerful demonstration of the intricate and often challenging dance between fungi and other life forms. They remind us that even within the most wondrous kingdoms, there are forces that challenge and reshape the biological landscape. And then there are the less common, yet equally fascinating groups that fill out the fungal kingdom. The Cryptomycota, for instance, a relatively new discovery, are minute, often single-celled organisms that reside in aquatic environments, representing some of the earliest diverging fungal lineages. The microsporidia, once thought to be protozoa, are now recognized as highly specialized, obligate intracellular parasites of animals, including humans, with a drastically reduced genome and a unique polar tube for infecting host cells. They are a testament to the fungal kingdom's ability to adapt to even the most extreme and intimate of parasitic lifestyles. As we survey this breathtaking panorama of fungal life, from the flagellated chytrids to the majestic Basidiomycetes, from the ubiquitous yeasts to the hidden Glomeromycetes, a profound truth emerges. The diversity of the fungal kingdom is not merely a collection of interesting forms, it is the very engine of their ecological power. Each phylum, each species, each lineage has honed a particular set of strategies, a unique biochemical toolkit to thrive in its chosen niche. This incredible variety allows fungi to colonize virtually every habitat on Earth, from the deepest oceans to the highest mountains, from scorching deserts to frozen tundras. It enables them to decompose nearly every organic substance, to form partnerships with almost every plant, and to regulate populations across the animal kingdom. It is this unfolding tapestry of forms, this symphony of specialized adaptations, that makes fungi the silent, indispensable architects of our planet. We stand at the edge of this vast, intricate world, having only just begun to chart its contours. What new species await discovery? What untold biochemical wonders lie hidden within their mycelial networks? What ancient wisdom can we glean from their patient, persistent existence? These questions beckon us forward, inviting us to continue our journey, to delve deeper into the mysteries of this profound and wondrous kingdom. For in understanding the fungi, we come closer to understanding the very pulse of life itself. The questions linger in the quiet air, don't they? What new species await discovery, hidden in plain sight or in the deepest, most inaccessible corners of our world? What untold biochemical wonders, what ancient wisdom still lie dormant within their silent networks, waiting for us to listen, to observe, to understand? As we draw closer to the end of our foundational journey, we find ourselves not at a conclusion, but at the threshold of an even grander exploration, one that delves into the profound potential of this kingdom, not just as an ecological cornerstone, but as a silent partner in shaping our future. Imagine, if you will, the vastness of the unknown. While we have journeyed through the established phyla, the familiar forms and the recognized roles, the truth is that the fungal kingdom remains largely a mystery. Scientists estimate that for every known fungal species, there could be anywhere from 5 to 10 or even 50 yet to be formally described. We are, in essence, peering through a small window into an immense, dimly lit chamber, catching glimpses of an unseen tapestry of life. Think of the rainforests, the deep oceans, the arid deserts, and even the seemingly sterile environments of our own homes. Each harbours a unique mycological community, a miniature universe of forms and functions awaiting our gentle inquiry. This immense undiscovered diversity represents a living library of biological innovation, a trove of genetic blueprints and biochemical pathways honed over eons, each one a testament to the fungi's unparalleled adaptability and resilience. We are only just beginning to learn how to read its ancient script. And what might this ancient script reveal? As we venture beyond mere classification and ecological understanding, we begin to uncover the astonishing ways in which fungi can actively contribute to a more sustainable, healthier future for humanity and the planet. This is where the wisdom of the mycelial network truly begins to unfold in ways that can profoundly impact our modern world, offering elegant biological solutions to some of our most pressing challenges. Consider the realm of medicine, where fungi have already gifted us one of our most revolutionary discoveries, penicillin. But penicillin, a marvel though it is, represents just a single note in a symphony of potential. The fungal kingdom is a prolific producer of secondary metabolites, complex organic compounds not directly essential for growth, but often crucial for survival in competitive environments. Many of these compounds possess potent biological activities, acting as antibiotics, antifungals, antivirals, immunosuppressants, and even anti-cancer agents. We are continuously sifting through this natural pharmacy, isolating new compounds from obscure species found in remote soils or deep-sea vents. Imagine the next generation of antibiotics, desperately needed in an era of rising resistance, emerging from a humble soil fungus, or perhaps novel therapies for neurodegenerative diseases, or entirely new approaches to treating cancers, all derived from the intricate chemistry of fungal life. The patient, silent intelligence of the mycelial network holds secrets that could redefine human health. Then turn your thoughts to the environmental crises that loom large. The fungi, those tireless decomposers, offer potent solutions in the field of bioremediation. Their enzymatic prowess, honed over millions of years to break down complex organic polymers, can be harnessed to dismantle pollutants that plague our soils and waters. Picture vast networks of specialized fungi deployed to clean up oil spills, their hyphae weaving through the contaminated earth, systematically degrading hydrocarbons into less harmful compounds. Envision fungi breaking down plastics, those seemingly indestructible symbols of our modern excess, slowly returning their constituent elements to the earth. They can even absorb and sequester heavy metals from contaminated sites, effectively filtering and purifying our environment. This isn't science fiction, it is the quiet, persistent work of the fungal kingdom, offering us a living, self-replicating technology for ecological restoration, a profound act of planetary healing. Beyond remediation, the fungal kingdom offers us pathways to sustainable materials and energy. Imagine a world where our homes are grown, not built. Mycelium, with its incredible tensile strength and ability to bind particles, can be cultivated into durable, lightweight, fire-resistant and biodegradable materials. Myco designers are already creating mycelium-based bricks, insulation, packaging, and even furniture. Materials that are not only sustainable but can be grown from agricultural waste products, turning what was once considered trash into treasure. This isn't just about replacing existing materials, it's about a paradigm shift, a move towards myco-architecture, where our built environments are in harmony with natural cycles, designed to eventually return to the earth without harm. Furthermore, the fungal capacity to break down tough plant biomass, like cellulose and lignin, makes them invaluable in the production of biofuels, offering a more efficient and sustainable route to energy independence, lessening our reliance on fossil fuels. We are learning to collaborate with these silent architects, allowing their natural processes to sculpt the future. In food and agriculture, the dialogue with fungi becomes even more intimate. Beyond the culinary delights of mushrooms like shiitake and oyster, we are exploring mycoprotein as a highly nutritious, protein-rich alternative to animal products, offering a sustainable solution to global food security. In the fields, mycorrhizal fungi are already indispensable partners, enhancing nutrient uptake and drought resistance in crops, reducing the need for chemical fertilizers and pesticides. But imagine further. Fungi as biological pest control agents, offering targeted, environmentally benign solutions to protect our food supply. Or even the development of crops that are more resilient, more productive, and more nourishing, thanks to a deeper understanding and strategic application of their ancient fungal alliances. And then we arrive at a more subtle, yet profoundly impactful realm, the exploration of consciousness and well-being. For millennia, various cultures have understood certain psychoactive fungi not merely as plants, but as sacred teachers, tools for introspection, healing, and spiritual insight. While the scientific understanding of these substances is still in its nascent stages, modern research is cautiously exploring the therapeutic potential of compounds like psilocybin found in certain magic mushrooms. Under carefully controlled clinical conditions, these substances are showing promise in treating conditions such as severe depression, anxiety, PTSD, and addiction, by promoting neural plasticity and helping individuals reframe their perspectives. It is a sensitive area, demanding profound respect and rigorous scientific inquiry, but it speaks to a deeper connection between the fungal kingdom and the very fabric of our minds, hinting at ancient wisdom that transcends the purely physical. We are learning, slowly and carefully, to listen to these quiet teachers, not for recreation, but for healing and a deeper understanding of our inner landscapes. As we ponder these immense possibilities, we must also consider our responsibility. The profound potential of fungi underscores the urgent need for their conservation. Many fungal species are highly specialized, forming intricate relationships with specific plants or animals, or thriving in unique microclimates. Habitat destruction, climate change, and pollution threaten countless unknown fungal species, each a potential key to a medical breakthrough, an environmental solution, or a vital link in an ecosystem's resilience. Protecting fungal biodiversity is not merely an academic pursuit. It is an act of safeguarding our own future, preserving the genetic and biochemical libraries that hold untold solutions to the challenges ahead. We are not just observing nature, we are stewards of its intricate dance. So as we bring our foundational exploration to a close, we realize that the journey into mycology is truly endless. It is a continuous unfolding, a slow, quiet conquest of understanding that reveals profound principles of existence, interconnectedness, resilience, transformation, and a delicate yet immense beauty. The fungi, often overlooked, often misunderstood, stand as silent sentinels of wisdom, patiently waiting for us to truly see them. What new discoveries will tomorrow bring? What ancient wisdom will we unearth from their silent networks? The mycelial web continues its patient expansion, a testament to life's enduring creativity. Our role, as curious observers and respectful partners, is to continue listening, learning, and imagining the possibilities that unfold beneath our feet in the deepest forests and in the quiet corners of our own lives. We are, all of us, part of this grand, ongoing conversation with the hidden kingdom, and the most exciting chapters are still waiting to be written. We have journeyed through the unseen tapestries of the mycelial kingdom, marvelling at its ancient wisdom, its profound ecological impact, and its boundless potential to shape our future. We've spoken of medicines yet to be discovered, materials yet to be engineered, and environmental solutions that lie dormant within the fungal genome. But as we reflect on these grand possibilities, let us also turn our gaze to the present, and indeed to the long, winding path of human history, where the fungal hand has been at work, often silently, often invisibly, shaping our cultures, our sustenance, and even our very sense of well-being. Imagine, if you will, the dawn of human civilization, a time before written records, before the meticulous classifications of science. Even then, we believe, our ancestors encountered fungi, not as abstract biological entities, but as tangible forces in their daily lives. They saw the sudden emergence of mushrooms after rain, a mysterious abundance from the earth. They observed the decay of fallen trees, the transformation of raw ingredients into nourishing food and drink. This wasn't science as we know it, but an intuitive, deeply ingrained understanding of the natural world, an awareness of the profound principles embedded in life's most unassuming forms. Consider first the most fundamental of human needs, sustenance. For millennia, fungi have been an integral part of our diet, gracing our tables in countless forms. Beyond the familiar cap and stem of the edible mushroom babur, the rich earthy flavour of a shiitake, the delicate texture of an oyster, the robust heartiness of a portobello, lies a vast culinary universe. These are the fruiting bodies, yes, the ephemeral blossoms of the unseen mycelium, offering their concentrated nutrients and unique umami to our palates. We cultivate them in controlled environments, coaxing them from carefully prepared substrates, a testament to our enduring partnership with these organisms. The slow, patient growth of the mycelium beneath straw or sawdust, its silent conquest of the substrate, culminates in that wondrous burst of form and colour we so eagerly anticipate. But the fungal contribution to our food goes far, far deeper than the visible mushroom. Think of the staff of life itself, bread. Without the tireless work of Saccharomyces cerevisiae, common baker's yeast, our loaves would remain flat, dense, and unyielding. It is this microscopic Ascomyceae, patiently consuming sugars and releasing carbon dioxide, that leavens our dough, creating that airy, inviting texture and complex flavour. And what of the beverages that have brought communities together for millennia? From the ancient fermented drinks of our ancestors to the modern craft beers and fine wines we savour today, yeasts are the silent alchemists, transforming simple sugars into intricate arrays of flavours and aromas. We invite them into our vats, a deliberate act of co-creation, allowing their metabolic processes to unfold, crafting liquid poetry. Even in the realm of dairy, where milk becomes cheese, fungi play a pivotal role. The distinctive blue veins of Roquefort, the creamy rind of Brie, the complex flavors of countless aged cheeses. These are often the handiwork of specific molds, primarily species of Penicillium. These molds aren't merely decorative, they actively break down fats and proteins, contributing to the texture, aroma, and taste profiles that define these culinary treasures. They are master sculptors of flavor, working their quiet magic within the cheese cave, transforming the simple into the sublime. And in Asian cuisines, consider the foundational role of Aspergillus oryzae in producing soy sauce, miso, and sake, a mold that initiates the complex enzymatic breakdown necessary for these staple fermented foods. It is a testament to the fungi's remarkable biochemical versatility, their abilities transform, to create, to enhance. Beyond the kitchen, fungi have long been revered for their medicinal properties. While modern pharmacology has isolated compounds like penicillin, the wisdom of traditional medicine systems across the globe has recognized the healing power of certain mushrooms for centuries. Reishi, Ganoderma lucidum, known as the mushroom of immortality in Eastern traditions, has been used to support immune function, reduce stress, and promote longevity. Lion's mane, Hericium erinaceus, with its cascading icicle-like spines, has been valued for its potential to support cognitive health and nerve regeneration. Turkey tail, Trametes versicolor, a vibrant, fan-shaped polypore, has been studied for its immune-modulating compounds, particularly in the context of cancer support. These aren't mere folk remedies, they are the culmination of generations of observation, experimentation, and reverence for nature's pharmacy. We are now, with our advanced scientific tools, beginning to understand the complex array of bioactive compounds, polysaccharides, triterpenes, glycoproteins, that confer these therapeutic effects. It is a beautiful convergence of ancient wisdom and modern inquiry, a recognition that the profound wisdom embedded in these organisms has always been there, waiting for us to listen more closely. And what of the deeper, often unspoken cultural significance? Across diverse cultures, fungi have appeared in folklore, myth, and ritual, sometimes as symbols of transformation and rebirth, sometimes as harbingers of mystery, sometimes as bridges to other realms of consciousness. The ephemeral nature of the mushroom, its sudden appearance and disappearance, its connection to both decay and new life, has imbued it with a potent symbolism that resonates deeply with the human psyche. They remind us of the cyclical nature of existence, the constant interplay of life and death, growth and dissolution. In our modern era, our interaction with fungi has become even more sophisticated, moving beyond traditional uses into the realm of industrial biotechnology. The very enzymes that fungi use to break down tough plant matter are now harnessed to our advantage. Fungal enzymes are crucial in detergents, making our clothes cleaner, in textiles softening fabrics, in biorefineries, converting biomass into biofuels, the citric acid that adds tang to our sodas and candies, the statins that help manage cholesterol levels, even the immunosuppressants vital for organ transplants. Many of these are produced through large-scale fungal fermentation. We have learned to cultivate specific fungal strains in vast bioreactors, coaxing them to produce the very molecules that underpin vast sectors of our economy and healthcare. This is not a passive relationship, it is an active, dynamic partnership. We provide the conditions, the nutrients, the careful stewardship, and in return the fungi offer their unparalleled biochemical ingenuity. It is a testament to their adaptability, their resilience, and their willingness, it seems, to collaborate. However, with this deepening relationship comes a profound responsibility. As we venture into the wild seeking edible treasures or medicinal marvels, The importance of accurate identification cannot be overstated. The beauty of a mushroom can mask a potent toxicity. The difference between a gourmet delicacy and a deadly poison can sometimes be a subtle shade of gill or a slight variation in stem structure. We approach the wild fungal kingdom with a blend of awe and caution, understanding that its gifts are accompanied by lessons in humility and meticulous observation. Foraging is not merely gathering, it is a mindful engagement, a quiet conversation with the forest, demanding respect and knowledge. And just as we recognize the value of individual species, we must also embrace the larger imperative of mycodiversity conservation. We spoke in Chapter 7 about the vast numbers of undiscovered species, the genetic library waiting to be explored. But this library is under threat. Habitat destruction, climate change, and pollution diminish fungal populations just as they do plant and animal life. To protect the forests, the soils, the very ecosystems we depend upon means protecting the unseen mycelial networks that underpin them. It means recognizing fungi as keystone species, as essential threads in the immense unseen tapestry of life. We cannot truly conserve a forest if we only focus on the trees, ignoring the patient, silent intelligence that connects them all beneath the soil. Our journey through mycology fundamentals is, in essence, an ongoing dialogue with a kingdom of profound wonder. We began by peering beneath the soil, discovering the vastness of the mycelial network. We explored its diverse strategies for life, its macroscopic impact, its microscopic architecture, and its intricate dance of reproduction. We classified its major phyla and glimpsed its potential to heal the planet and ourselves. Now, we reflect on its enduring presence in our daily lives, from the bread on our tables to the medicines in our cabinets. This relationship, ancient and ever-evolving, calls for a new level of appreciation and stewardship. It asks us to look beyond the immediate, to perceive the interconnectedness of all things, to recognize the profound wisdom embedded in life's most unassuming forms. The fungi, in their patient, silent work, continue to teach us about resilience, transformation, and the subtle power of connection. And as we continue our slow and thoughtful adventure, we carry with us the understanding that this hidden kingdom is not just a subject of study, but an indispensable partner in the grand unfolding story of life on Earth. What new secrets, what new collaborations await us in the chapters yet unwritten? We can only approach with open minds and a sense of profound wonder, ready to learn and ready to appreciate, ready to participate in the ongoing conversation with the fungal world. We have journeyed together through the vast hidden kingdom of fungi, peering into its ancient wisdom and marveling at its intricate designs. We have seen how these silent architects underpin the very fabric of life, from the grand cycles of decomposition to the intimate embrace of mycorrhizal partnerships. Their resilience, their quiet intelligence, their profound interconnectedness, these qualities have filled us with awe, reminding us of the immense and often unseen forces that shape our world. Yet even the most ancient and resilient beings are not immune to the pressures of a rapidly changing planet. As we deepen our understanding, we must also confront a sobering truth. This vital kingdom, so often overlooked, is facing unprecedented threats, and its silent cry echoes through the very ecosystems it sustains. Imagine, if you will, the immense subterranean web of mycelium stretching beneath our feet, a delicate yet powerful network that breathes life into the soil and mediates the flow of nutrients across continents. Now, imagine this web being torn, fragmented, or poisoned. The consequences, we are coming to understand, are not merely local, they ripple through the entire biosphere, affecting everything from the health of our forests to the stability of our climate. One of the most immediate and pervasive threats to fungal biodiversity is habitat loss and fragmentation. As human populations expand and our footprint on the Earth intensifies, natural landscapes are increasingly converted for agriculture, urban development, and infrastructure. Forests, which harbour an astonishing diversity of fungal species, are felled at alarming rates. Wetlands are drained, grasslands are ploughed, and even remote wilderness areas are encroached upon. For fungi, whose existence is so intimately tied to their specific substrates, a particular tree species, a decaying log, a patch of undisturbed soil, the destruction of these habitats means the destruction of their very homes. When a forest is clear-cut, it is not just the trees that vanish. The entire mycorrhizal network that sustained them, the saprophytic fungi that recycled their nutrients, and the myriad other species that formed an integral part of that ecosystem are also lost. The mycelial highways are severed, the ancient connections broken, and the profound principle of interconnectedness is violently disrupted. Consider the delicate balance of a forest ecosystem. Each tree is a testament to the profound partnership it shares with its fungal allies. When these trees are removed, the fungi that form mycorrhizal relationships with their roots, or the arbuscular mycorrhizal fungi within their cells, face immediate collapse. These fungi cannot simply relocate. Their spores may drift on the wind, but without suitable host plants and the specific environmental conditions they require, their chances of re-establishing are slim. What remains is a fragmented landscape where isolated patches of mycelium struggle to survive, their genetic diversity diminished, their ability to adapt compromised. We are witnessing, in essence, the unravelling of an unseen tapestry, thread by precious thread. Beyond direct habitat destruction, climate change presents a multifaceted challenge to the fungal kingdom. Fungi, like all organisms, are exquisitely sensitive to environmental conditions. Shifts in temperature, altered precipitation patterns, and increased frequency of extreme weather events, droughts, floods, wildfires, can profoundly impact their life cycles. Many fungi rely on specific temperature ranges for fruiting, spore dispersal, or the successful establishment of their mycelial networks. A warmer climate might push some species beyond their physiological limits, while others may find their symbiotic partners, be it a specific plant or insect, struggling to adapt, thus breaking crucial ecological links. We are observing changes in fruiting seasons, shifts in geographical ranges, and even the emergence of new fungal diseases as pathogens adapt to altered conditions. The delicate dance between fungi and their environment, honed over millions of years, is being thrown out of sync, and the consequences for ecosystem stability are deeply concerning. Then there is the silent, insidious threat of air pollution. Our modern world, with its relentless industrial activity and extensive agricultural practices, releases a vast array of chemicals into the environment. Fungicides designed to protect crops from pathogenic fungi inevitably impact non-target species, disrupting the beneficial fungal communities essential for soil health. Herbicides, while targeting plants, can indirectly harm fungi by altering the vegetation composition or through direct toxicity. Industrial pollutants, heavy metals, and microplastics permeate our soils and waterways, creating toxic environments where fungi struggle to thrive. These substances can inhibit fungal growth, impair their metabolic process, or even lead to genetic mutations, weakening their ability to perform their vital ecological roles. The very ground beneath us, the cradle of the mycelial web, is becoming a hostile landscape for these foundational organisms. We also face the challenge of invasive species. In a globally connected world, the accidental or intentional introduction of non-native fungi, or the pathogens they carry, can devastate native ecosystems. A novel fungal disease for which native species have no evolved resistance can sweep through populations with catastrophic speed, much like Batrachochytrium dendrobatidis has decimated amphibian populations. Conversely, invasive plants can alter soil chemistry and microbial communities, making it difficult for native fungi to persist. These biological invasions disrupt the existing complex web of interactions, leading to a loss of native biodiversity and a homogenization of ecosystems. Perhaps the most profound threat, however, lies in our collective lack of recognition and research. For too long, fungi have occupied a mycological blind spot in conservation efforts. While we meticulously document and strive to protect charismatic megafauna and rare plants, the hidden kingdom often goes unnoticed, unlisted, and consequently unprotected. Our understanding of fungal biodiversity is still rudimentary. We estimate that only a fraction of fungal species have been formally described. How can we protect what we do not even know exists? This knowledge gap means that countless species may be vanishing before we even have a chance to discover them, taking with them their unique genetic resources, their specific ecological functions, and their untold potential for medicine, bioremediation, and sustainable solutions. The absence of comprehensive fungal surveys, the scarcity of mycologists in many regions, and the limited funding for fungal research all contribute to this perilous oversight. The consequences of fungal loss are not abstract, they are profoundly real and impact us directly. We are speaking of the potential collapse of nutrient cycling, leading to less fertile soils and reduced agricultural productivity. We're talking about forests that become more vulnerable to disease and climate stress without their mycorrhizal partners. We are envisioning ecosystems stripped of their primary decomposers, where organic matter accumulates, carbon sequestration is disrupted, and the very breath of the planet is stifled. The unseen tapestry, once vibrant and resilient, begins to fray, threatening the stability of the entire biosphere. But this is not a narrative of despair, but rather a call to stewardship and enlightened action. Just as we have embarked on this slow and thoughtful adventure to understand fungi, so too must we embark on a collective journey to protect them. The first and most crucial step is education and awareness. We must shatter the mycological blind spot, bringing the wonders and vital importance of fungi into the public consciousness. By understanding their roles, appreciating their beauty, and recognizing their vulnerability, we cultivate a reverence that naturally leads to a desire for protection. Research and mapping are equally critical. We need dedicated efforts to discover, describe, and understand the vast diversity of fungal life. This means funding mycological expeditions, supporting taxonomists, and utilizing advanced genetic techniques to unravel the complexities of fungal communities. Citizen science initiatives where enthusiastic amateurs contribute observations and specimens can play a powerful role in expanding our knowledge base, mapping distributions, and identifying areas of high fungal biodiversity or particular concern. Conservation initiatives must move beyond a focus solely on plants and animals to explicitly include fungi. This involves protecting fungal habitats, particularly old-growth forests and undisturbed soils, through the establishment of reserves and the implementation of sustainable land management practices. Reducing the use of broad-spectrum fungicides and herbicides in agriculture and forestry, and exploring more targeted, ecologically sensitive approaches is paramount. We must also invest in bioremediation strategies that can mitigate existing pollution and restore contaminated ecosystems, often harnessing the very power of fungi themselves. Finally, we must advocate for policy and legislation that recognize fungi as essential components of biodiversity, granting them the same consideration and protection afforded to other kingdoms. Listing threatened fungal species, supporting their inclusion in environmental impact assessments, and integrating fungal health into broader conservation strategies are vital steps. As we conclude this chapter, let us reflect once more on the profound wisdom embedded in life's most unassuming forms. Fungi, with their intricate interconnectedness and their tireless work behind the scenes, offer us a powerful lesson in interdependence. Their silent cry for protection is not just for themselves, it is a plea for the health of the entire planet, for the delicate balance of life that sustains us all. By becoming conscious stewards of the mycelial web, by recognizing its value and actively working to preserve its integrity, we are not only safeguarding a kingdom of profound wonder, but also ensuring the resilience and vitality of our shared home. The journey of discovery continues, and with it our responsibility grows, inviting us to partner with these ancient architects in building a more sustainable and harmonious future. We have journeyed together, you and I, through the intricate wonders of the fungal kingdom, peeling back layers of misconception and revealing the profound, silent intelligence that underpins so much of life on Earth. We have seen the ancient mycelium as the unseen architect, the diligent recycler, the vital partner, and the quiet healer. Our exploration has led us from the microscopic dance of hyphae to the macroscopic majesty of ancient forests, all connected by an immense subterranean web. And in our most recent reflections, we grappled with the pressing threats that loom over this essential realm, understanding that the survival of the fungal kingdom is inextricably linked to our own. Now, as we near the horizon of our shared adventure, we turn our gaze towards the future, a future not merely of passive observation, but of active, conscious partnership with these remarkable organisms. The call to stewardship, which resonated through our previous discussion, is not a burden, but an invitation. It is an invitation to redefine our relationship with the natural world, to move beyond a paradigm of extraction and control, and to embrace one of mutual respect and collaborative wisdom. For too long, the fungal kingdom has languished in a mycological blind spot, overlooked by science, marginalized in conservation efforts, and often misunderstood by the public. This oversight is not benign. It is a profound deficiency in our collective ecological literacy. To truly protect and partner with fungi, we must first learn to see them, not just the fleeting fruiting bodies, but the vast patient intelligence of the mycelium itself. This requires a fundamental shift in our perception, a broadening of our ecological empathy to include these silent essential beings. We must cultivate a sense of wonder that extends beyond the charismatic megafauna and vibrant flora, reaching into the delicate, complex beauty of the unseen world beneath our feet. Imagine, if you will, a world where fungal literacy is as common as botanical knowledge, where children learn about the wood wide web alongside photosynthesis, where the quiet heroism of decomposers is celebrated. This future begins with education, in our homes, our schools, our communities. It involves fostering a deep curiosity, encouraging hands-on exploration, and making the invisible visible through accessible science and art. Citizen science initiatives, for instance, offer a powerful avenue for this transformation. When individuals are empowered to document local fungal diversity, to identify species, and to contribute data to global databases, they become not just observers but active participants in the grand unfolding story of life. They become advocates, their appreciation deepening with every new discovery, every mycelial network traced, every spore print taken. Beyond education, true stewardship demands concrete action in conservation and policy. Just as we protect ancient forests for their towering trees, we must recognize that their health is inseparable from the health of their fungal partners. This means safeguarding old-growth habitats, which harbour invaluable fungal biodiversity that cannot be replicated in younger managed forests. It means minimizing the use of broad-spectrum fungicides in agriculture and land management, understanding the collateral damage these chemicals inflict upon beneficial mycorrhizal networks and decomposer communities. It necessitates creating protected areas that specifically consider fungal habitats, recognizing that a fallen log or a patch of undisturbed soil can be a vibrant, irreplaceable fungal ecosystem. Policy, too, must evolve. We need legislation that acknowledges the ecological significance of fungi, integrating them into biodiversity conservation strategies and environmental impact assessments. Imagine a future where the health of the soil microbiome, teeming with fungal life, is a primary metric for agricultural sustainability, where urban planning incorporates mycorrhizals to connect fragmented fungal habitats, and where the economic value of healthy ecosystems includes the silent services rendered by fungi. This is not a fanciful dream, it is a pragmatic necessity for a resilient, thriving planet. And what of the future of human-fungi collaboration? We've already glimpsed the immense potential of fungi in medicine, bioremediation, and sustainable materials. But a future of true partnership goes beyond mere utility. It embraces a reciprocal relationship. We are learning to cultivate mycelium not just for its products, but for its inherent intelligence in crafting materials that are regenerative, biodegradable, and beautiful. Think of mycelium-based packaging that returns to the earth without a trace, or mycelial building blocks that breathe and adapt, contributing to the health of their surroundings. This is not simply using fungi, it is learning from their millions of years of evolutionary wisdom, allowing them to guide us towards more harmonious ways of living. In medicine, the discovery of new fungal compounds continues at a breathtaking pace, offering hope for combating antibiotic resistance, developing novel treatments for neurodegenerative diseases, and addressing mental health challenges. But our partnership here extends beyond the laboratory. It involves respecting the traditional knowledge of indigenous cultures who have long understood the healing power of fungi, integrating this ancient wisdom with modern scientific inquiry. It means approaching fungi not as a mere source of chemicals, but as complex biological entities whose intricate chemistries are a reflection of their profound evolutionary journey. Perhaps the most profound lesson fungi offer us as we look to the future is about interconnectedness itself. The mycelial network is a living metaphor for the intricate web of relationships that sustain all life. It teaches us that true strength lies not in isolation or competition, but in collaboration and mutual support. It reminds us that waste is merely a resource in the wrong place, that decomposition is not an end, but a vital transformation, and that even in decay, there is the promise of new life. Fungi embody resilience, adapting to changing environments, finding pathways around obstacles, and patiently, persistently transforming their world. They are masters of the slow, quiet conquest, a testament to the power of persistence and the wisdom of living in harmony with natural cycles. As we conclude this chapter of our exploration, We acknowledge that our understanding of the fungal kingdom is still in its infancy. For every species we identify, countless more remain undiscovered, hidden in the depths of our oceans, the canopy of our rainforests, or the very air we breathe. The unseen tapestry of profound wonder continues to unfold, revealing new mysteries with every step. The fungal kingdom is an inexhaustible source of fascination, a living library of solutions, and a profound teacher. So let us carry forward this newfound appreciation, this sense of reverence into our daily lives. Let us look at the soil beneath our feet with new eyes, recognizing the vibrant, intelligent network that thrives there. Let us advocate for the silent partners of our ecosystems. Let us remember that we are not separate from nature, but an integral part of its grand, interconnected design, just as a mushroom is but a fleeting expression of a vast, enduring mycelium. The journey of understanding fungi is an ongoing one, an invitation to remain curious, to remain humble, and to continue listening to the profound wisdom embedded in life's most unassuming forms. What mysteries will you uncover next? What fungal conversation will you overhear in the quiet moments of reflection? The mycelial future awaits, and we are together its architects.